And we know that those droplets are physically unstable, so we need to stabilize them with an emulsifier. And those can be surfactants, so very low molecular weight molecules absorbing at the interface. These can be particles that have already been widely studied in non-food related research, but there's now also a trend where people really are trying to find food grade particles. But we can also use proteins, and these are actually very widely used in the foods because they are natural, so we can put a natural claim. They work very well, and they also add to the nutritional quality of the food products. So they have a lot of advantages. And then if we look at the proteins that we now currently are using, these are mostly dairy-derived proteins, so coming from a cow, which could be whey protein isolate or sodium caseinate, and you might know them from the sports drinks. But whereas in Delft, we really talk about the energy transition to get more sustainable world. In Wageningen, we are very much focused onto the so-called protein transition. And this basically means that we're trying to go from animal-derived proteins towards plant-derived proteins for sustainability issues. Well-known plant protein is, for example, soy protein. But now an emerging protein is also pea protein isolate. Because peas have the advantage that we can also grow them in Europe. There has already been some research done, and pea proteins seem to work quite well, and plant proteins in general seem to work quite well, but their functionality is still lacking compared to the dairy-derived proteins, not only in terms of functionality of stabilizing the interface, but also, for example, in the taste that they have. And therefore, we thought in my PhD that it might be nice to combine the two and actually use blends of the two proteins. To kind of compromise between functionality and product quality, and also have a little bit more sustainability. I already talked a lot about the interface, and during my whole PhD, I really was very much interface focused. And this is because actually in emulsions, it's an interface dominated system. So if we emulsify 1 gram of oil, there's more than 10 square meters of interface present. So you can imagine that this interface is very important for all emulsion characteristics. Then if we use proteins, and especially protein blends, we get a very complex composition at the interface with a high structural heterogeneity. And on top of that, actually those interfaces evolve over time. So there's aging, the proteins are unfolding, maybe connecting with each other, but also together with the bulk, there might be adsorption and desorption over time. So it's a very non-equilibrated system. And in the beginning, we realized that, well, it could be nice to use those protein blends, but it would be important for us to understand what actually happens at the interface. And we had in mind that 4 different things could occur. We could get a very homogeneous interfacial layer, where both proteins contribute to the interface stabilization. But it could also be that it doesn't work that well, and that actually the proteins start to phase separate at the interface. And then you could actually get weaker spots next to the phase separated domains. It could be that one protein is so surface active that it completely dominates the interface. And then the second protein would adsorb on top of this, and we would get kind of multi-layer formation at the interface. Or it could be that the proteins would be not compatible at all, and only one protein adsorbs at the interface, whereas the other one remains non-adsorbed in a continuous phase. And in all those cases, well, different phenomena would occur. So we decided to really look at those interfaces of the emulsions through the full life cycle of the emulsion. So the life of an emulsion basically starts when they are produced. So within a homogenizer, the oil is broken up into smaller droplets and the initial interfaces are formed. And then after emulsion formation, the emulsions, of course, are stored and aging occurs. And then we were wondering what would be most relevant in every stage of this process. And we realized that, or we know very well that during homogenization, it's very important that those proteins absorb immediately at the interface. And this really happens within sub-second scale. So for this, we decided to use microfluidics because we can measure at very short time scales what happens at the interface and with the emulsion droplets. We were also very interested in the strength of those interfacial layers and the structure, because this could explain why some droplets remain stable or become unstable. And this we did with so-called model interfaces. For example, a drop tensiometer has been widely used. But also the composition of the interfacial layers is very important to know which protein actually contributes to the stability and to the network formation. So we also did some direct analysis of the emulsions, and then we tried to combine all those different length scales and time scales to each other. And from this, we could really understand what happens from the subseconds towards the weeks, even months time scale. So I would like to start explaining what we do with the drop tensiometer. And we use this to quantify the interface's mechanical properties up to hours. And you probably know a drop tensiometer, but very shortly what we did is we created a small oil droplet in a watery protein solution, and then we capture the shape of the droplet, and based on the shape we could calculate the interfacial tension. And the first measurement we do is always a time sweep, where we keep the area of the droplet constant. And because the proteins absorb, the interfacial tension will actually decrease. And I think this method has been widely used in many different research fields. But what we also do quite a lot for protein stabilized interfaces is dilatational rheology, because we also want to know more about the strengths of those networks formed. So what we do is, if we have the droplet, we make it bigger and smaller in a sinusoidal way. And then we measure the interfacial tension response. And then based on this, we can calculate the elastic and loss moduli of the interfaces. So for surfactants, it's very normal to do frequency sweeps, where you always keep the same amplitude, but vary the speed in which you do the oscillation. But for these viscoelastic properties, it's also very nice to do amplitude sweeps, where you keep the same amplitude, or where you keep the same frequency of your sweep, but increase the amplitude over time. So these are my results for the whey protein isolate. So we see the elastic moduli as a function of the applied amplitude. And we see that at low amplitudes, we have actually a very stiff interfacial layer with a high elastic modulus, around 30. But what we see is, if we increase the amplitude, we see that the elastic moduli actually decreases as a result. So there's quite a clear strain dependence present in this elastic moduli. And this is really an indication of the breakdown of interfacial protein aggregates due to the large amplitudes that we apply. If we look at the pea protein isolates, we also see quite a stiff interfacial layer with an elastic moduli around 20, but there is no strain dependence. So the network is less affected by the applied amplitude. If we then look at the whey pea protein blend, so 1-to-1 blend of the two proteins, we see that the elastic moduli is actually very similar to the pea proteins, but we cannot really say a lot about it yet. So this is actually the basic information that you can get, but these data points actually have much more information inside them. And this is because those data points are actually the average from what happens if you expand and compress the interface. But it would be much more useful if we could actually see what happens throughout the full cycle with the interface. And this we can do with so-called Lissajous plots, especially in bulk rheology. They are very well known, but now more and more people also use them for interface rheology. And basically what you do is you plot the change in surface pressure as a function of the change in area. And a completely elastic interface would then give a linear line. A very viscous interface would give a spherical line. And if you have something in between, a viscoelastic interface, you kind of get this ellipsoid shape of your Lissajous plot. So by doing this, we can really see what happens if we go from compression towards extension and then back to compression again. So we did this also at the oil-water interface. And you can see that overall for the whey protein isolate Lissajous plot, we see a clear viscoelastic behavior, which is well known for proteins. But we also we can see that there are some non-linearities present. For example, we see here that the slope of the Lissajous plot actually becomes lower. And this is typical for strain softening behavior. Or you could also say that there is some weakening in extension. We can also see that if we compress the droplets and subsequently extend it again, that there's kind of a spherical shape here in the Lissajous plot. And that basically means that if we compress the proteins at the interface, that they really start to interact with each other, and there's a certain strain needed to expand again. So we really can see that there's a kind of network formation here. If we look at the P protein isolate stabilized interface, we see that the elastic properties are quite different. We even see much stronger strain softening in the extension, so you really see a flattening of the curve. But we also see this very interesting pointy shape in compression and subsequent extension. And that basically means that if we compress the proteins together and then expand again, that there's nothing, that nothing happened at the interface. So the response from compression and extension is exactly the same, which shows that there were no interactions induced by concentrating those proteins at the interface. And we actually see the exact same for the whey P protein blend. So also there we only have very weak interactions. And what we think is that in the protein blend, actually both proteins are present at the interface, but the P proteins really hinder the whey proteins to form this nice interconnected layer at the interface. So now we knew a bit more about the strengths of the interfacial layers. So we decided to look at the emulsion stability. So I created emulsions with a high pressure homogenizer. And at day 0, we see that all the emulsions had the same droplet size. And we were able to create very small droplets. But when looking over after 14 days again at our emulsion, I saw that my P protein stabilized emulsion had a second peak, indicating that larger droplets existed within the emulsion. So this could be that either the droplets coalesced, so they really merged together, or the proteins or the droplets were flocculated into small clusters, which would then also be detected as larger droplets. And to differentiate between these two mechanisms, we simply dilute our emulsion in an SDS solution. And SDS can replace the proteins from the interface and then absorb themselves at the interface. And because they're very negatively charged, protein or emulsion clusters would then break up again into their individual droplets. And we actually saw that for the P protein stabilized emulsions, after dilution in SDS, we got a monodispersed emulsion again, showing that we only had flocculation in those systems. And we did the same for 3-to-1 and 1-to-3 blends, and we got the same results. So those emulsions remain stable. So we believe that those P proteins, well, they may be flocculated more because there's quite some hydrophobic interactions between the interfaces, because plant proteins are much more hydrophobic compared to the dairy proteins. And if we add some whey proteins in the mixtures, then we have more electrostatic repulsions that overcome those hydrophobic interactions. And we also believe that because those P proteins still were able to form a stiff interfacial layer, even if there's not a lot of connection, they still had a high elastic moduli. This was strong enough to protect the emulsion droplets against real destabilization via coalescence. And this is why only flocculation occurred. But of course, if you work with those blends, we needed to verify that actually in the protein blend stabilized emulsion, both proteins adsorbed at the interface. So I checked the composition of the interfaces, and I did this over time. And I saw that every time if I used a blend, so ranging from 3-to-1 up to 1-to-3, always both proteins are present at the interface, but actually the composition of the interface changes over time. So I saw, especially in the 3:1 and the 1:1 ratio, that over time more and more whey proteins were present at the interface, and they actually displaced the P proteins. So the P proteins were displaced from the interface towards the continuous phase. And then I was thinking what the reason could be that I see this in the two upper systems, but if there's less whey proteins present, then I do not see this anymore. And I hypothesized that this could be because there's simply less whey proteins already initially present at the interface. So maybe then it's more difficult for an extra whey protein to also absorb at the interface. Or it could also be that we, in the continuous phase, need a certain amount of whey proteins for this displacement to occur. So I really made a full new set of experiments. And what I did is I first made an emulsion with just one protein, so that I have an interfacial layer consisting of one protein. And then I add the second protein post-emulsification. And I made emulsions where I already mixed the emulsifiers before, the two proteins, to create a mixed interfacial layer. And then I added the second protein post-emulsification. And I summarized the results. So if we start with the whey protein stabilized layer, and I add P proteins to this layer, I did see that a lot of the P proteins absorbed, so we actually got higher surface loads, but over a 3-day period, no displacement occurred, so the interface composition was actually fixed. If I started with P proteins, so P protein-based interface, and I added whey proteins to this, I again saw that the whey proteins absorbed immediately at the interface, and over a period of 3 days, the concentration of whey proteins actually increased, and there were less and less P proteins present at the interface. So it was clear that in this system, we already saw displacement, even if the whey proteins were not initially present at the interface. And then if we had the mixture, if I added P proteins, nothing happened over time. Whereas if I added whey proteins, again, I saw displacement of the P proteins by the whey proteins. So it was clear that the whey proteins needed to be present in a high enough concentration in the continuous phase to be able to displace the pre-absorbed P proteins. And of course, you may think like, why is this so interesting? Well, because if we would start with a P protein-based interfacial layer, we know that flocculation occurs, but then if over time, if we just add whey proteins, the emulsion can become stable in this case, but maybe in other cases, the emulsions can also become destabilized because we compromise the interfacial layer because the structure simply changes upon the sequential introduction. So again, we looked into this using a drop tensiometer, but now we changed the experimental design a little bit. So we first let one protein absorb, and we did a little test sweep to test the strength of the interface. And then we did a bulk exchange. So we exchanged 5 times the volume of the cuvette with the other protein solution. And then we let this absorb for 1 hour, after which we tested the rheological properties. And the results are very interesting. So these are the interfacial tensions that we measured. So I started here with the P protein isolate, and we see that the interfacial tension decreases. Then if I do the bulk exchange and I add the whey proteins, I actually see an increase in the interfacial tension when we include the whey proteins in the system. And if we start with a whey protein isolate solution and we add the P proteins, we see a strong decrease in the interfacial tension due to the addition of the P proteins. And then looking at the elasticity, because that will tell us a bit more about the connections that are present. We see that if we add P proteins, there's actually a strong decrease in the elastic moduli. So it goes from 30 for just whey proteins to around 15 if we add P proteins as a second layer. So the P really compromises the whey protein stabilized layer. And we see the opposite. If we have a P protein stabilized interface and we add the whey proteins, we see that the elastic moduli increases, and we also see that this strain dependency is there again, showing that there's a bit more connection in the interfacial layer. So now we were wondering what are then actually the reasons for the displacement? Because if you think logically, interfacial tension would always be most favorable to have the lowest interfacial tension in your system from a thermodynamic point of view. But we actually see that the opposite occurs. So the whey proteins tend to be at the interface, even if this leads to an increase in the interfacial tension. So we believe that actually those in-plane protein interactions that the whey proteins are able to have actually favors this displacement and actually lowers the total energy of the system. So this was very interesting overall, because we could really show that if you use those protein blends, we can actually play with the structure of the interface. So even if we have the same overall composition, dependent on how we add the proteins to the system, we get very different interfacial structures. And we should not forget that they also age over time. So their properties are never the same at different time points. So this was all about the long-term stability of the emulsions. But I also looked in what happens during the formation of emulsions. And as I said, this happens in a homogenizer at very, very short time scales. So if we have enough proteins or other emulsifiers present, the newly formed droplets are immediately stabilized, and we will get very small droplets in our final emulsion. Whereas if we have not enough emulsifiers present, the emulsions will immediately recoalesce, leading to larger droplet sizes. And we know that for standard emulsifiers, one of the most important things they do during the homogenization is to reduce the interfacial tension. But from the long-term experiments, we also know that proteins form this viscoelastic interface at longer time scales. But we do not know if this also already happens within the homogenizer or if it's just interfacial tension effects that play a role here. And basically the reason why we do not know this is because if you want to do rheological measurements, you typically do this with a tensiometer or maybe a Langmuir-Trapp. And those measurements are simply not fast enough for rheological properties. So you can measure the interfacial tension after, well, let's say half a second, but rheological properties, you need to do the full sweep. So the times do not really overlap. So this is why in the last 2 years of my PhD, I really focused on using microfluidic chips to link the stability of droplets to coalescence to the interface rheological properties within the same time scale. And at this time, this would really be the sub-second scale. So first we used a quite standard chip that we widely used already before in our lab. And what we do is we create very monodisperse droplets using protein solution as the continuous phase. And then those droplets move through a membrane channel where the proteins can further absorb. And then the droplets enter a coalescence channel. And then we measure the droplet sizes at the beginning of the channel, and we measure it also at the end of the channel. And based on the area change, we can determine how often a droplet coalesced. And if we look at the results for the proteins, and we see here the number of coalescence events as a function of the protein concentration, we see that the lower the protein concentration, the more coalescence we get, which is as expected. But we also see that the concentrations needed to stabilize the whey protein layer or whey protein droplet are much lower compared to the plant proteins, since we need much more proteins. And this is because the pea proteins are actually quite aggregated compared to the dairy proteins. And from more non-food related research, we know that if you have a Pickering system, that actually bridging between the two droplets by aggregates can induce coalescence. So we believe that at low surface coverage, the bridging by the protein aggregates actually induces coalescence. So we need more proteins to really stabilize the interface. If we then look at the one-to-one blend, we see that this is actually not the average behavior of the individual proteins. but that the behavior is much closer towards what we expect or towards what we measured for the P proteins. So to compare this a bit better, we determined how much proteins we need to get a coalescence of 0.4. And we did this for various ratios. So you can see that to get a coalescence of 0.4, you need around 0.25 grams per liter of P proteins, whereas we only need 0.03 grams per liter of whey proteins. So again, highlighting that whey proteins are much better at stabilizing the droplets. But we also see that if we only add a very small amount of P proteins, immediately we need much more proteins in total to stabilize the interface. So from this, it's clear that at those short time scales, the P proteins actually hinder the whey proteins a lot in interface stabilization. So as you can already guess, based on the content of this presentation, we were again interested in the rheological properties of the interfacial layer and what the effect of the P proteins was on the interface formation. So then we developed a new chip within my project, where again, we created droplets using T-junction, but then the chip design looks a bit different. So the droplets enter the main channel and then they are accelerated by another protein solution so that we could really look at individual droplets. And we push those droplets through constrictions. And those constrictions were around 160 milliseconds, 0.5 seconds, and 1 second. And based on the deformation of the droplets through the constriction, we could extract rheological properties. And those are really at very short time scales that were never measured for protein stabilized interfaces. So some basic theory about the droplet deformation. This is of course dependent on the viscosity ratio between the dispersed and the continuous phase, which we kept constant in our experiments. It's also dependent on the continuous phase viscosity and the droplet velocity, which we again kept constant. And it's dependent on the interfacial tension. And then you would expect that if you have a lower interfacial tension, you actually get a higher deformation. So this is well, some of the basic. And then we were looking at our experiments. And we started with the whey proteins. I plotted here the maximum deformation as a function of the whey protein concentration. And we actually see that if we have a higher protein concentration, our deformation is lower. So based on a simple theory, this would mean that the interfacial tension is higher in this system compared to this system. Well, we know that this is not true. So these are actually the rheological properties of this protein interfacial layer that actually give a resistance against deformation already after 160 milliseconds. And this is probably because of the van der Waals interactions and hydrogen bonds between the proteins. We see more or less the same trend for P proteins, but we see that there's first an increase in the deformation, probably because more proteins absorbed, reducing the interfacial tension. And only at higher concentrations, there were enough proteins present to resist the deformation and therewith decrease the maximum deformation. And also for the whey P protein blend, we saw the same trend. So the maximum deformation decreased as a function of the protein concentration. So overall, these experiments are already quite insightful because we can see that, well, the P proteins are much less capable of resisting deformation at those short time scales, and whey proteins form the stiffest interfacial layer already at those time scales. But there's much more information that we can get out of these measurements, because now I only showed you the maximum deformation, which we see as a measure of the stiffness of the interfacial layer. But we can actually also look at the relaxation time. So the time it takes for the emulsion or for the droplet to go from its deformed state back to its spherical shape. And this tells us something about the viscous and elastic contribution. And then an elastic interface would go back immediately to its spherical shape, whereas a viscous interface would remain deformed for longer. And to extract those relaxation times, we used a stretched exponential, including a beta. And we needed this beta because we know that protein stabilized interfaces are actually very heterogeneous, especially upon deformation. So it's very important to include this beta factor. If we then looked at the deformation and the relaxation, we see that the whey proteins had the highest relaxation times, indicating the most viscous interface, whereas the pea proteins were most elastic at the first constriction. But we did the same after 1 second, so at the last constriction in our chip. And actually we saw that in all cases, the relaxation times increased. So in all cases, there is a higher viscous contribution of the interfacial layer. And very interestingly, we see that if we use a protein blend, the relaxation time is actually highest. So we expect that over time, we actually have viscoelastic property changes due to rearrangements at the interface already within 1 second. And we also think that if both proteins are present, we actually have some enhanced adsorption, which gives the most viscous interfacial layer. And we saw the same trend for the other concentrations. So to conclude this microfluidic part, we could actually determine for the first time that interfacial elasticity already plays a role in the sub-second range for those interfaces. The whey proteins have the highest resistance against deformation, and they have a very fast interfacial film formation, and therefore they're also very stable and well, have a low coalescence. Whereas for the pea proteins, well, they were not at all stable to coalescence. We needed a lot of proteins. And this is probably due to the low resistance against deformation and the high concentrations and time needed to develop the interface. Whereas with the blend, the rheology chip showed us that we have enhanced adsorption and film formation over time. But we needed 1 second for this, and this is not fast enough to protect against coalescence. So this would be more important for the long-term stability. Yeah, so we had really some new insights for protein stabilized interfaces, that we could show that there's a viscoelastic layer, that the interfaces behave like heterogeneous solids at those time scales, and that the relaxation time and the maximum deformation could give us rheological properties. Then some overall conclusions about protein stabilized interfaces. Well, again, we showed that elasticity plays a substantial role in the sub-second range, but we also show over longer time scales that the interfaces really show nonlinear behavior upon deformation. And one of the major parts that I looked into is that the interfaces are really non-equilibrated system that change composition and properties over time, something that is quite often overlooked for protein blend systems. For the whey proteins, we saw that the film was formed very fast, already within 0.16 seconds. And after 3.5 hours, we got a very interconnected network with covalent bondings inside that really gave a high physical stability to the droplets. And we also well, had a crossover point where we went from a viscous dominated to an elastic dominated interface. The pea proteins absorb slower, but over time they could still form a stiff interfacial layer, even if it was less interconnected. And for the protein blends, we saw that there was an increased stiffness after 1 second, probably because of enhanced adsorption. And we could also measure this in the surface loads of the emulsions. But over the longer time frames, yeah, it's clear that the PPI hinders the network formation of the whey proteins. And actually the whey proteins then also start to displace the pea proteins. They were changing the interfacial properties. So this was everything I wanted to tell you about my PhD project. If you have any questions regarding this project or maybe also about my postdoc, feel free to send me an email. And of course, you can also ask them now.
Thank you very much, Emma. And my compliments for keeping the talk very accessible by all the time switching between the results and the conceptual and even molecular picture behind them. So what I would like to do is actually open up the floor for questions. So if you have any question for Emma, please open up your microphone
Can I ask Emma? Very nice presentation. Can you hear me?
Yes.
I still, very interesting result, which you showed the compression curve from the Lisa-Jones plot was different from the stretch extension. Am I correct? That's something.
Yeah, the nonlinear behavior of the interfaces.
Yeah, I was just wonder how much effect of gravity will influence that behavior. Is it important or no? In your
We make the droplets larger and bigger.
Yeah, that's during the because you are using like a oscillatory type of the. There is always gravitational force which can enhance compression compared to extension because the force is oppositing. Is it relevant or is not relevant? Again, I'm not expert on this behavior.
Well, you need to use the to determine the interfacial tension from the shape. You need to have certain criterion to be met. So also the size and actually the shape of the droplet always needs to be Laplacian in order to do this. So as long as you do this, I think it's negligible.
Or maybe let's put it, if you put the reference material, which has a nonlinear response in the extensional compression, you will see symmetric behavior.
You mean, sorry, what do you mean?
So the droplet, which doesn't have too much protein, or the which is causing these that's what I'm understanding from your curve. Extension against compression has a different response. Did you check this setup with the reference material, which I don't know, just a simple viscoelastic?
Yeah. So
droplet?
for if you do it for example, SDS, you get a completely elastic. It's like a straight line.
Oh, perfect. Okay, perfect.
Thanks, so this is quite typical for protein stabilized layers.
Yes. Okay, clear. Thanks.
Thanks, Pouyan. Are there perhaps any other questions or comments for Emma?
Maybe I have one question.
Please.
Like in your experiments, you always looked into pea and whey protein. And what do you think, how much can you like generalize on also other proteins, like soy protein or other alternatives?
Quite a lot. And this is mainly because there's a big problem they are now facing is I was using a commercial plant protein isolate. And it's very difficult to isolate those plant proteins. So they're highly denatured. And basically they are all present as small aggregates. So yeah, I also have some work done on this, which I did not include in today's presentation. So it's not really a typical plant protein. It's more a commercial plant protein aggregate, I would say. But I think like the general insights that we obtained, so for example, when does this displacement occur and why does it occur? Those insights are general for every system, not even only for plant dairy, but also just for dairy systems, because we should also realize that a whey protein is not just one protein, but also contains already 4 types of proteins. And a plant protein typically contains 6 to 7 kind of proteins. So we never work with pure protein systems, never.
Okay, thank you. Maybe like just a really short question, like which would be what would be applications where you use both plant and dairy protein, because the dairy seem to work better. So if we want to, I don't know, make a vegan product or so, we just use pure plant.
And then you cannot use the dairy.
No.
Well, for example, in quite some products that are dairy-based, they now add extra dairy proteins, and they could decide to replace part of those dairy proteins by plant proteins. Also, cost-wise, until now, plant proteins were a bit cheaper, but I guess this will change soon. But for example, they already do it for the tube food. It's already a combination of plant and dairy proteins.
Thank you.
Can I ask a small question about the application of your work? You mentioned on a certain moment a colloid mill, and I was wondering whether the quantities or the measurements you have made on a very microscopic scale, especially the mechanical properties, how they contribute to understanding how a colloid mill works and whether you can use it in the food industry.
Yeah. Actually, I compared the results. So even if it's model systems, they do give some insights. So actually the results that I got with the microfluidics actually also match what I found with a real homogenizer. So I saw that if there's not enough proteins present, for example, if I use 0.28% pea protein or whey protein, I get much smaller droplets with the whey protein compared to the pea protein, even if I use the same homogenization pressure. So I think it gives a lot of insights, and especially the advantage of microfluidics is that those timescales really match the processes in industry, even if they're probably much more energy efficient, and you even have more, well, of course, also turbulence playing a role in homogenizers. But I think for now it's a very nice method, also because it's not purely diffusion based, the interface formation. So it's really bridging the gap between model interfaces and emulsion characterizations.
Okay, thank you.
If I'm not mistaken, there was also a question by our other Peter. Am I correct?
Well, I switched on my video. Yes, I have a question. Thanks for the very interesting lectures. A part of the questions have been covered by looking at the applications. I'm part of the PDN group where we do product design, chemical product design, and I was wondering what kind of products would you aim at? Would you aim at the mayonnaise with some vegetable protein in there? And well, would this method then be needed, or can it be used at these high concentrations of oil, or would you simply do trial and error like most people in industry do?
Well, I as a researcher would never really go for trial and error, I guess. But no, for now, I really, this work was all done on dilute emulsions. And even if it maybe sounds a bit applied, it was quite fundamental for food research. So it's really a few steps ahead from direct applications, I would say. But actually now with Volkert, I'm looking into more concentrated emulsions. And there actually plant proteins work very well, especially dependent on what you want. If you want to have a high rheological properties or like a very viscous emulsion, then plant proteins can actually help you a lot. Whereas, for example, if you use dairy proteins, they are much more fluid-like even at high oil concentrations. So also in those type of systems, you can play around with the macroscopic properties by changing the properties.
Now, what I was wondering next to that is, you talk about pea proteins, like it is one variable, but I'm sure that when you, for instance, treat those proteins with higher temperatures or whatever, that the properties will drastically change. So then you get a whole set of variables next to it. Are you going to treat that?
Well, I'm not working anymore with plant proteins. But no, this is a very big problem. And also I use just one batch of plant proteins because they are like, there's already so many batch to batch differences. And also the plant proteins, if we extract them from the plants, we have a lot of non-protein components, such as lipids, phenolic compounds that affect the interfacial properties. So I think we are really now at the start of trying to understand how those plant proteins work. And we are now building slowly by very slowly the knowledge. But it's really a whole new research line that is now hot topic in the food world.
So you cannot get pro-analysis plant proteins. It's
Sorry?
You cannot get them, say, pro-analysis, so that you have a very well-defined plant protein that would require complete development.
Well, I have one paper written about this. I did not include it here, but there I used very purified proteins, but I only had 1 gram and it took 10 weeks to make it. So you're not going to put this in a homogenizer.
No, then you need to develop a process that makes this, and that's going to.
Well, or maybe we need to. I think this is now maybe more the trend that we are just going to accept that maybe the plant proteins work in a different way than dairy proteins, but we start to use the proteins that we can use for a certain application rather than trying to directly replace them. I think that would be much more sensible to see the strengths of the plant proteins and the strengths of the dairy proteins and use them where they would fit that.
Yeah, the dairy proteins originally were developed to make milk, to do emulsification. So
yeah,
that's
to
why
provide
they're so
nutrition.
suitable.
Yeah.
Okay. Thanks.
No problem.
Okay, thank you very much, Peter. I don't see any raised hands. Is there perhaps there is opportunity, of course, for a question or a comment now, but if nobody uses that opportunity, then I take the opportunity to once more thank Emma. Yeah, I see Lucas joins me in that in thanking Emma. So. As I mentioned at the start, please see the presentation by Emma, which was on proteins, really as an invitation on maybe further discussions on interfaces in general. Maybe you don't work on proteins, maybe you work on nanoparticles or macromolecules in the solution that also happens to contain interfaces. And maybe there are things that you always wanted to know more deeply, but didn't have a chance to look into. I think Emma has beautifully shown us that just doing, say, a pendant drop or a static interfacial tension measurement tells you something, but perhaps not at a time scale relevant for the formation that actually happens in your synthesis or in your process. So thank you all for being here. Have a happy Monday, and see you next week. Thanks, Emma.
Thank you.